I have my colleagues here, John preparing our soil samples for the microscopy, and Melissa is preparing samples for the microbiometer. These are the two tools that we're going to be using today to demonstrate how to evaluate soil microbes. Very briefly, when we're talking about soil microbes here, we are talking about specific groups within the soil community. There's this thing called the soil food web, where it talks about the different trophic levels that exist in the soil. These are categorized by, first and foremost, our plants that are actually photosynthesizing, taking all that beautiful sun rays, capturing those and making very complex substances such as, or known as exudates, right? So they send all these exudates, they're very complicated molecules, but essentially sugars down to the roots. These sugars in turn actually attract a series of microbes, specifically fungi and bacteria. There's a bunch of genera, species that we won't get into. What we are looking for in this case are, again, morphological features. We are looking at the shapes. Nobody needs to understand what these are doing, how they're functioning. It doesn't matter. What we really need to be paying attention is diversity. And so how do we figure out whether or not we have this diversity? We can actually see that through the microscope. We can see shapes, right? We can see if it's circular. We can see if it's elongated. We can see colors. So these are the type of things that we're going to be using to identify the microbes by morphological in a morphological way. And so we're not going to be counting, or that's why we actually have the microbiometer. The microbiometer is going to give us a count of total biomass. It'll also give us a bacteria and fungal ratio. It can't discern whether or not those are the types, but we are not going to be paying attention to those things because, quite frankly, it doesn't matter. What really matters is that we actually see different things under the soil. If you're going out to be a researcher and you want to do actual papers, then that's probably something that you may want to get into, of learning what these things are and what they're doing. Word of caution, and this is something that I learned from a Mexican microbiologist. He mentions how, and I like to use John for this example because I've done this talk before. So let's say I take John and I put him into a laboratory, and all I do is I feed John beans constantly, and John just eats up all the beans, right? He's just eating beans all day, all night. And me as a scientist observing John, I say, well, look at John. All he eats is just all he eats. That's all he eats. He eats, just consumes beans. And so I made a reductionist approach to this. I gave him this food, and then I made the assumption that that's all that John eats. And so we have to be careful when we're actually talking about these microbes, of their function and what they eat and what they consume, because quite frankly, there's way too many. Most of them are not culturable, that we can't culture in a lab, and a lot of them are actually working in function. And so when we actually take one microbe, we may make an assumption and say, well, this microbe does this. But in reality, when it's out in the field, it may have a consortium of different microbes that are actually functioning together. And so we have to be careful how we make these assumptions. So, which is why I go back to my point, we can measure diversity by just looking at shapes, colors, and sizes. Any questions about soil food web?
When you talk about trophic level,
yep.
Does that mean, like, on a really, really basic sense, if you look at the soil and you see some big organisms, that means there's enough little organisms to feed those big organisms, so your soil's healthier? It's probably not that simple.
Yes, exactly. There's a little more nuance to that. However, that's a good proxy still, right? So if you actually have, let's say, predators in any ecosystem. Let's not even talk about soil right now. Let's talk about the jungle. And in your jungle, you actually have a mouse, you have you have your rodent, you have a big cat or something like that. That big cat can exist if that rodent's not there, right? But that's generalizing too, because that cat may actually eat other things as well. And that cat, to what Dan was talking about earlier, to that point of, is our microbiome healthy? Just because we're standing here doesn't mean that we have a healthy microbiome, right? And so we can't make that assumption that that cat is healthy because it's eating a diverse diet, because we see it, right? But it could be used as a proxy. But we have to be careful with generalizing too. However, still a good sign, right? If you see predators in your soil, that means that the soil is functioning as an ecosystem, that there's nutrient balancing, that there's actually nutrient cycling happening as well. Amongst those soil trophic levels, I mentioned bacteria and fungi. There's also protozoa, which are the predators of the soil. Those are classified in two major groups: the ones that actually live in high oxygen conditions, the ones that live in low oxygen conditions. And so if you move into the next trophic level, you will then find nematodes. Nematodes come in different varieties. These are actually bacterial feeding, fungal feeding, root feeding, which is the ones that we are always scared about and don't want in our soils. And then there's the predatory nematodes, which actually consume others. They apparently, those predatory nematodes tend to actually like the more pathogenic ones. We don't know why, but that's some of the stuff that has been discovered. So that's a basic overview of the soil food web. We're looking for maximum diversity. We are looking for, and as we put samples onto the screen, we'll also talk about the different features of the soil too. It's not just in the microbes, but what the microbes are doing. Ultimately, you can take this information. We're digging deep into the soil, but if we were to go outside and dig a hole and we pull out some, let's say a root, and this root is covered in a rhizal sheet, it's just fully covered, we know that this plant is interacting with microbes. We don't need a microscope for that, right? What we are using the microscope for is looking at that diversity. Again, to your point earlier, just because something is there doesn't mean that it's actually diverse. It may be interacting with the soil, it may be functioning at a certain level, but these tools can actually provide us a better picture of whether or not we are actually seeing this diversity. And who are we missing, and why are we missing them too, right? So those different trophic levels are the ones that we're going to be looking for, and the ones that we want to encourage in our soils too. As I mentioned earlier, John is preparing some of those slides there for us. We will call on all of you who brought your soils or compost in to talk a little bit about this. But what we're going to do is we're going to start putting some samples onto the screen and just making general observations about what we're seeing. We'll confirm then with you guys who brought the soils in to see, are we on point or are we completely off target here? So let's just get started with some of the soils.
Where's the best place to buy a microscope from? Relatively inexpensive.
Well, you can join NOFA Mass as a member.
Okay.
And you can actually take advantage of some of the discounts that we offer for our members.
Okay.
There's discount codes for LW Scientific.
Okay.
That is not the best entry-level microscope for people that are wanting to get into this. And so if you're looking for just a entry-level microscope, AM scope, or OMAX,
okay, are
definitely microscopes to look at. We're not going into like high magnification here.
Right.
For the
400.
Yeah, for the most part, we're just going at 400x. Okay. So you don't need any more than that. All right. So right here we're at 4x magnification.
not a lot to talk about here. This is actually at the lowest magnification we have. We will go in and go in a little deeper into this. But generally speaking, what you're seeing here is just, sparse aggregation, maybe some aggregates here, and not much to talk about there. Let's go ahead and go for 100x total magnification. Again here, we're seeing very similar features. Probably now at this magnification is a good time to actually go ahead and scroll through to see what we find. All right, so that's the edge of our slide there. As you can see, there's a little bit of water on the other side there. So we're just going to quickly scroll through here, see if there are any large microorganisms. Some of those that we just discussed earlier with the trophic levels, specifically nematodes. You sometimes can see arthropods as well, but I am not seeing anything yet. That is an air bubble, and that is at the edge of our slide again there. That is a nice little piece of aggregation there. Let's see if we can find anything else. Not a lot going on here. I'm not seeing any filamentous organisms, like something that you would find like as a little stick. Sorry if I'm actually making anybody dizzy here. Here we go. Let's actually zoom in into this area here so we can talk a little bit about what we're seeing here. So I'm going to switch over to 400 total magnification. And this is where you can actually start to see bacteria. There's the smallest round circles that you see, like those there, here. Those will be your bacteria. Now, there's a thing called Brownian motion. Just because they're moving around doesn't mean they're actually alive or active. It could just be that, this is happening. And so whenever you're seeing movement, don't get that mixed with something that's alive or active. It could just be a simple effect of Brownian motion. You'll see when these microbes want to move because they move very intentionally. And hopefully you can find one that is actually moving other than the bacteria that are here. A few things that I wanted to talk about on this particular slide are these little microaggregates. These microaggregates can only form if we actually have the microbes present. These are fungi and bacteria that create glues. These are the things that are going to hold your soil together. The more of those microaggregates you have, the more water stable compounds will form in the soil. Your nutrients will stay in the soil, less leaching, similarly with water as well. So whenever you're looking under your microscope, that is one thing that you want to be seeing. Another thing too that you want to see is you want to make sure that you can see through your sample. You don't want to see the soil too dense because all that means is that your sample is actually not holding itself together. Another thing too that I wanted to point out was this guy over here. That looks like an egg to me. I'm not sure of what organism that belongs to, but it looks like an egg. It could be a nematode egg. Anything else in there, guys, that you can see that looks interesting?
So what are the things, what is actually moving and what is vibrating in the water in terms of that picture right now?
Yeah, and that is 100% hard to tell unless you had a stain. So if you have a stain that can tell you whether your bacteria are active, you can then say, okay, well, those are bacteria, they're active, and they're really moving. when you're looking at this, you can't tell what's alive, what's dead. Another thing too that nobody really talks a lot about is that you can't tell archaea you can't distinguish archaea nor bacteria. Not this way. You would need a DNA analysis to actually figure that out. They're completely indistinguishable when you're looking at them under the microscope.
What you can do, though, I mean, the bacteria tend to be symmetrical, and you're either going to have, dots or rods, as opposed to all the soil bits that are erratic. And most organisms have some symmetry to them, as opposed to plant material and mineral material. This table is, this is all shaking a lot.
Yeah.
We're on a,
Plastic.
Yeah.
Folding table.
But when you, and if you're making teas and extracts, particularly teas, when you're feeding these, the movement's insane.
Okay.
When you give them food and get them going, it's not this Brownian motion that we're seeing anymore. You'll see, they're moving, and it becomes very apparent. You can have these little clusters, swarms of bacteria, little cloud happening all in itself.
But that usually happens when the soil's been in water and doing...
In a tea, actually,
In a tea,
where we've
okay.
taken compost, put it into a brewer, probably given it some molasses.
Gotcha.
That's when everything starts coming alive. And compost. Soils, like, under the microscope, are typically the most boring things that we look at. Good quality compost, you can see a lot of activity.
Oh, no wonder.
Going back to Dan's presentation this morning, it's pretty apparent that most of our soils are beat up. So and it correlates with all of their research because most of the soil that we look at ain't that exciting.
Can we know a little bit about the context of the sample?
Yeah, let's actually zoom into this thing right here because it may be interesting. So, what you're seeing there is a protozoa. This is actually considered a arcella, and it is a predator that we just discussed earlier. If you have this organism there, that means that it's actually probably consuming bacteria. It will consume bacteria. Every organism in the planet has a carbon to nitrogen ratio. All that means is that all that excess nitrogen that it consumes will be pooped out, and it's going to be in plant available form. There's this thing called rhizophagy cycle that also takes place. So there's two main ways that the plants will get their nutrition from: through the rhizophagy cycle, which is them taking microbes inside of their roots and cycling those nutrients from the bacteria, and then there's whatever's in the solution. Whatever's in the solution is what poops out of these microbes. So those are the two main ways that plants mainly get their nutrition. I would say, just to make general comments about this, seeing a protozoa here, there's definitely some aggregation happening at different stages of aggregation. So the darker colors that you can see here versus the more honey color-like, those will indicate the different types of humic acids that are in there. And as you know, this humus is actually formed by further decomposition, right? So you have organic matter decomposition, and then you just have further decomposition past that. You can't decompose further than this humus, and that should be our ultimate goal. Our ultimate goal should be, how are we humidifying our soils? And those are great indicators of such humification too. There's another protozoa.
But you see,
Yeah, okay, that was my question.
There is another protozoa up here. This is actually a testate amoeba. So amoebas are actually, it's like a loose term to describe a bunch of different protozoa within this class. But it's a very complex family. There's other features in here that are somewhat not popping up. that I do want to bring up are this filamentous guy here. You can probably see it over here as well. Those are a type of bacteria, believe it or not, and also known as actinobacteria. Somebody was asking about triangles. Are you talking about the crystal-like?
Yeah, that guy right there.
That's just sand.
This is silica.
Yep, silica, exactly.
Can you get a sense from looking at this about the bacterial or fungal ratio of this?
That we're going to leave to the microbiometer.
But yeah, you can.
But yes,
time,
absolutely.
looking, because we haven't seen
Much fungus.
Right.
like next to none.
Exactly. If we were to look at this and say, okay, well, what are we missing? We know that we're missing fungi. And so how do we actually try to increase that fungi? This goes back to the principles that Dan was talking about earlier. They're very, very basic principles of how to manage soils.
There really weren't many protozoans in there to speak of either. Were there?
Well, this is actually 1 gram of soil and a drop from that gram of soil. So I would say as long as you say 5 of them, and we didn't go through the entire slide, we saw 2.
Okay.
So I would say that the protozoan population is actually active, meaning that they actually have enough food sources as well. And they were different, right? We saw an arcella, and we also saw a tested amoeba, both amoeba, but we saw 2 different types. So we saw some diversity, we saw some humidification happening, and a lot of bacteria, obviously. Well, the one thing that we're missing are perhaps fungi, and also we didn't see any nematodes either. So we want to, so we're missing 2 groups in here. Right. So I'm actually priming them in here. Gotcha.
You do also need to be cognizant about bringing food sources in for, that fungal population, and that to, mulches, plant material, leaving things around for them to, consume. But there's a reason why the weed is there, because of this. Because this is where you're getting your nutrient cycling. These microbes are making things available for your plants, and that diversity is going to dictate what plant is actually happy in that soil. It's fun, because what happens when you start changing these things, what you'll end up seeing is, to Dan's point about, pests, once you start moving these soils to be a little bit more fungal, you'll see that your pigweed is the plant that's covered with bugs, because it lost all of its partners, so it doesn't have the ability to get the nutrition anymore, so now it's susceptible to plant and disease. And it starts falling apart, and plants that are, more successionally developed are flourishing because they now have the community that's supporting them.
All right, so this is the other sample that we have here. I'm just going to zoom in really quick because I saw some movement over here. That's interesting there. So what you're seeing there is a type of amoeba. This is a naked amoeba. So the ones that we saw previously actually had some shells around them. They create these shells from their environment. So whatever's in their environment, they would create that shell. This one is really happy being in its environment. Whenever you see a naked amoeba, it's a great indicator that the soil conditions are optimal for its growth. It's not forming a test. It's not forming a shell to protect itself. So it probably has proper moisture, probably enough food sources, and other environmental conditions are just ripe for it. So that is a predator that we're actually seeing. Let's see.
It's a predator, but also that thrives in aerobic conditions.
Those exactly. So we still haven't seen anything that thrives in low oxygen conditions. that just looks like some plant matter, actually. However, that there... Oh, something going on down here. This is perhaps actually another protozoa, but hard to tell. Sometimes they get shy when you're looking at them. This one's getting a little camera shy here. Oh, here we go. So this filamentous organism that you're seeing there, that is a fungi. This is something that we didn't see in the previous soil sample, something that we want to be seeing all throughout, really, into the soil, and as much as we can. The more melanin that you see in, meaning the more color that you see, it's typically a sign of a more beneficial fungi. So we are seeing more here and here. Again, this is great to see. Again, the aggregation that we're seeing here, another great sign that the soil is aggregating. I couldn't tell if that was actually on the slide or...
So this A3?
This is A3, yes. So we're seeing more fungi here. This is at 400 total magnification.
That guy on the right, is that a protozoan?
Nope, that's just another mineral particle. You can see how it's, you know, almost translucent.
Now, how can you tell that that's a mineral particle and not the amoeba that we were looking at earlier?
I would say from experience, once you start looking at these things, you'll actually start to pick them up. It's kind of like when, you know, if you're a botanist in training, you know, you first start seeing green everywhere until you actually start to meet the plants and say, oh no, that's yarrow, or that's a catalpa tree, or, you know, and to the untrained eye, yes, everything looks green and it's all plants a plant, but once you start looking at these things, and it's only really a handful of things that we're looking for either. So it's not like we're looking for, you know, tens of thousands of these things, which is why I said there's only those few groups that we're concerned about.
Is there a good, like a Peterson guide to soil microscopy that you can identify different visuals on?
There's some resources out there. Microbe Organics, run by Tim Wilson. He has a good library of microbes.
Monique's actually putting together. Monique's our colleague from CT NOFA, and because of the microscopy class that we're doing, she's generating lots of charts.
Okay.
So Food Web does have like a poster too as well. I don't know if that's still in circulation, but you know, pictures, images.
Okay.
I will say, like going back to the symmetry, that big crystalline structure that you were like, that looks like a testate. It was not quite as symmetrical as, I mean, the testate amoeba are oval, but there's still some symmetry there.
Okay.
And most of the microbes that we're looking for have a level of symmetry. Even like the filamentous organisms are going to be consistent in the width, whereas like a root hair will get fragmented and taper down and get big. And the only, I guess, exception to that is these things called omycetes, which are like your molds and stuff. They're just this mess of tangled fibers and little egg-like structures, and they get pretty crazy. But everything else, like that, at least what we're trying to, you know, identify as being beneficial, you know, is pretty uniform in their thing.
Right. Couple things here. Soil Food Web School does not tell you to count spores. The reason why they say that is because a spore can germinate tomorrow or in another 100 years. However, after speaking with Dr. Johnson, that created the bioreactor, he actually has indicated that he counts his spores because they are an indicator of that there's a population in the soil that can germinate. And so, but he's looking at it because with the lens of creating this highly fungal type of compost, which I highly recommend everybody gets into Johnson Su bioreactors. The reason why I bring that up is because there's two fungal spores here. There's this guy here and that there. We don't count them.
I think the microbiometer also captures the presence of
Correct. Correct.
part of the microbial.
Right. Yep.
And that there is, so depending on the test, because like microbiometer is actually looking at total microbes. So a food web, if you send your sample out and you get a soil food web test, you're only getting an indication of active microbes. So there is, and it's like everything, it's important to know what is the test testing, and are these, you know, when we're talking about biology, are they active microbes or inactive?
If I were to give this a score, I don't want to score things, you know, but if I were to give this a score, I would say that we, or just briefly talking about this or summarizing this particular sample, that's another protozoa there, perhaps a flagellate that consumes bacteria. This sample appears to actually have all the aggregation that we talked about, the different colors of aggregation. We're seeing bacteria, we're seeing fungi, we're seeing happy amoeba, and for the most part, well aggregated soils. So one interesting thing that happens with some microbes, some of them will encyst, and some of them actually will create some endospores. Protozoa seem to make a cyst when the conditions are not right for them. So this that you're seeing right there, that is a protozoa that's waiting for the right conditions to actually pop out. If we probably left this on the table here and measure it a little while, it'll probably come out of its cyst. But again, soil food web does not count that. It makes an observation of it. But important to make that observation, because if you have the microbes there and they're cysting, that you need to, that's an indicator for you to understand that you need to change the conditions in your soil to create the environment for them.
Is that a fungal hyphae to the right of it?
That looks like a fungal hyphae to me, actually, and maybe two separate ones, actually, just overlapping. Even though we put in a drop here, this is actually, if you look at it microscopically, there's a lot of water in here. And so that's why when you zoom in and out, you're going to see different layers of what we're seeing. So this is actually at the top layer. We can still see bacteria here, right? And if you zoom all the way in, probably there, you'll see different strata of the slide.
All this nutrition, if it's locked up in all these microbial bodies and nobody's eating anybody, the plant can't get that nutrition. Somebody needs to be eating, and that's what nutrient cycling is, really somebody eating somebody else and pooping out fertility in a plant available form. Because these bacteria and fungi are literally dissolving rocks, taking that nutrient into their body, and then when they get eaten and pooped out, that rock mineral form is now plant available. So that's mineralization. If you read about mineralization, it's making the parent material plant available. Plants can't do that. Plants can't get the mineral out of the parent material. They need these microbial partners to do that, particularly bacteria and fungi. That's how soil created. There was a time when this earth was nothing but a large rock, and then wherever the bacteria and fungi came from, they started dissolving this rock, and they filled soil. And then the plants were like, hey, we got something to eat. And then plants started on this planet. It has nothing to do with chemical companies.
And it's not really even the bacteria and the fungi, it's the protozoa poop after eating the bacteria and fungi.
And then this whole thing about endophytes and rhizophagy, there's this, the plant is also pulling in these bacterial bodies in the plant, which is relatively newish. And some of the plant physiology books have to be rewritten because they didn't address that. They were just talking about ionic exchange. But as it turns out, there's a whole slew of microbes in the plant, just like there's a whole lot of microbes in you. And those microbes are called endophytes. So something that's living within another organism that is actually beneficial is an endophyte. And some of Dr. White's work is showing that the plant is getting 30% of its nitrogen from bacterial bodies.
From within it or from external?
By taking in bacteria and then hitting it with superoxide, shucking the cell wall off of the bacteria, it's taking, because all these bodies have a carbon to nitrogen ratio, so that nitrogen is getting shucked off of the bacteria. The plant is utilizing it, along with who knows whatever fun, phytochemicals that are happening. And now you've got this whole chemical thing that's happening within the plant with yeast and bacteria. And then it ejects that bacteria right back out into the soil profile so that the bacteria can regenerate, and basically it creates this cycle right in the rhizosphere. And all they're really looking at right now is nitrogen, but think about everything else that is in this plethora of, bacteria and what's yeast doing inside the plant. We know what yeast does in our beer. The chemical concoctions that's happening, the plant's probably regulating all this because it's the plant. But it's right.
So this soil sample here, we're on to A5, and briefly speaking, this is actually at 100x total magnification. I'm not going to say anything, and I'm actually going to start asking some questions now that we actually have looked at about, 3 samples now? What are the first observations that we can make out of this sample right here? Actually, let me just show you as well something interesting about these. This is 1, 2, 3, 4, 5, and 6. So we're looking at sample number 5 here. Very different from the rest of them, right? Can you see the difference on the screen too?
It's silty.
Very silty, right?
And we're looking at that, and the other ones have settled out.
Yep.
And this one, the silt is still.
Yep.
Okay.
Yep. And so, but there's actually a couple things with that too, because it's not just the silt, right? Because silt will settle in about 5 minutes. That's been probably sitting there for about 5 minutes. Whenever you do the whole texture thing of sand, silt, and clay in the jar, your sand will settle immediately. The silt takes a little bit of time, and then your clay finally settles, right? But what we're seeing here on the screen is that, and let's zoom in for a second. What we're seeing on the screen is that this is pretty dense, isn't it? But it's not necessarily dense with microbes. It's just dense with all the parent material, right? To me, this appears that it's actually not aggregating enough. When you're looking at a sample, again, you want to be able to see through your sample. You don't want to see the sample too dense, because that, to me, is an indicator that the soil is not aggregating properly. And I could be wrong, but let's keep on looking in here and see what we find.
So will this not hold moisture?
Exactly. Exactly.
Is there not enough pockets in it?
Yeah, so there's
Or does that have to do with air too?
Yeah, so the moisture, the reason why it won't hold enough moisture is because that aggregation is happening from the microbial glues, specifically the fungi and bacterial glues that are holding things together, right? If you don't have those glues happening, which, by the way, protozoa and other predators stimulate them to create these environments because they want to hide. They want to create environment. So all this aggregation is actually happening because predators are stimulating them to actually create this environment for them. So if you're not creating this environment, you're not creating an environment where your bacteria and fungi are creating these glues to then actually create water stable compounds. and so you won't have that decomposition or decomposition from organic matter. And it's that organic matter that's really going to help you with that water absorption too. So along with that, nutrient too, like the nutrient holding capacity may be lower in this particular sample. You can see that it's actually very sandy too. That's a piece of aggregate there. I mean, I do have to say that there's a lot of potential in the soil, and maybe with time and enough care, this does have the potential to actually.
What's the biggest bacteria that you would see on there?
Actinomycetes, the one that I mentioned that was actually nice and long. Those can actually span pretty large.
So that one down at the bottom of the screen, that's not a bacteria. It looks like a bit. No, that would be a bacteria. The big rock.
Talking about this right here?
Yeah.
No, that's just organic matter.
Okay.
Yep.
But this could be.
Could that be a rod?
Yep, that's actually actinomycetes there.
Okay. And then all the dots are cocci.
Exactly. Exactly.
So actinomycetes is a fairly easy one to identify.
It is actually, and the nice thing about the actinomycetes, and one of the tricks that I use is that when you're going up and down and focusing, it will leave like a pencil line, and that's like a telltale sign. You're like, okay, that's the pencil line, that's an actinomycete. And they should be about 1 micron in size. The way you actually identify 1 micron when you're looking at this is by looking at a cocci. A cocci should be about approximately 1 micron in size. And so
a cocci is 1 micron, but actinomycetes?
It will be within width of 1 micron size. Right. What I can say about this compost is it's actually bacterially dominated, and we'll have to see what the microbiometer says. Understanding that the microbiometer also picks up some things that we can't see. So we have to be careful when we're comparing these two things. But, you know, in a compost, you know, you definitely want to see every trophic level. Your compost is supposed to be more alive than your soil, right? And so you want to see every single trophic level in your compost.
And the fungus usually will come later in the curing process.
It does. So the maturation phase... Oh, actually, let me backtrack on that, because that is the reason why I mentioned the Johnson-Su bioreactor. There are different types of compost that lend themselves to creating the environment for fungi. Something like a Johnson-Su bioreactor, actively aerated compost pile that's not turned. It's that constant turning, really, that's actually not allowing for the fungi to proliferate. And so if you actually have a system where you are just letting a compost in place, whether that's a moldering, whether that's static pile, whether it's a static aerated pile, with either passive or blown in air, you're not turning that. And so that's actually a great site for fungi to proliferate. This does look like a ciliate. Paramecium is probably a good guess. I'm not sure. But it is a ciliate. And it's moving slower than they usually do.
Yeah.
And that's just probably because of how how dense this soil is, right? What we're seeing here is another protozoa. The way it's moving, I can't tell what it is, but based on its movement of that bumbling motion and what John mentioned earlier about getting stuck, what's stuck is actually its flagella, and that's actually how it moves around. And it looks like it actually got stuck with some of the particles that are there. But that's another protozoa. I'm seeing a lot of spores in here, which goes back to my point earlier that this actually does have the potential, and maybe it just needs that resting phase. I do want to mention, since we're actually on a compost sample here, last couple of years we were part of a SARE grant, which is Sustainable Agriculture, Resource and Education, where we went around 5 different farms, different composting systems. One of them actually shared compost. And what we did, we actually took spent mushroom substrate, which is actually the waste that comes from growing edible mushrooms, specifically grain and sawdust. And so after a flush, which is called, it's called a flush when you actually get one bloom of your mushroom, you harvest that. Some people go through different flushes, others actually just discard that. This farmer in particular does not want any contamination, so they actually discard the mushroom blocks. Our experiment involved taking all these different mushroom blocks, mixing them together, and then mixing them with compost. What we found was, and this was actually mixing it with finished compost, what we found was that was enough to stimulate and invite more microbes into the party. And compost got really exciting after a couple weeks of incubation. We noticed the trophic levels increasing, specifically nematodes. And at the time of analyzing this, I was talking to my colleagues. I'm like, am I counting something wrong here? There's just no way I have all these nematodes in here. What we found later on by a professor at Clark University was that oyster mushrooms send out chemical signals into their environment to attract nematodes. They do this so that when their nitrogen sources, remember we talked about carbon and nitrogen source, right? Everything needs nitrogen. So they attract the nematodes to their surroundings so that when the nitrogen sources are low, they can then consume them. So this was just another strategy to actually increasing that biodiversity in your compost by just adding this waste product. We noticed that at least 25% was enough to do this. 50% was optimal.
50%?
Compost to SMS. Absolutely. Wow. Yeah. A couple things that we noticed and other things that we analyzed were electrical conductivity, which some people were like, oh, the salts, that's the problem, but that was not a problem with this type of mushroom growing. The salts are really more for like the agaricus, dung-loving mushrooms. These were more wood-loving mushrooms. So we analyzed EC, and we also analyzed pH. pH did drop in the compost, and the reason for that is because of the enzymes that fungi produce. In order to what John was saying earlier, in order for them to actually digest their foods, they digest their foods outside of their body, so they have to produce enzymes to break down these materials. So the pH did go down a little bit. I use spinach substrate to compost on my own, and I don't know, and I see a lot of nematodes, but I'm not sure if those nematodes are coming in after or before. Or do those compounds persist too? That's the other question. Are some of these compounds that are invisible to us, of how all of these microbes communicate, are they still in these compost piles? And so I argue that people should do different type of composting to get different results. So if you want more fungi, go ahead and do your bioreactor. If you want more bacteria, then, do your thermophilic and, don't let it rest for too long. There's a lot of biochemical reactions that we will not be able to capture under the microscope, but are way more important if we actually are looking at compost as an inoculum, as a biostimulant, because those chemical compounds are there. And again, the name of the game is bringing in maximum diversity. If we have maximum diversity, we know that we actually have a plethora of these chemicals in your compost, that they can then be used as, a top dressing or a compost tea, or, inoculating your seeds. As we know, seeds, the whole industry is a whole issue there. So, we need more inoculums. A point on organic matter, very, very important. and there's been studies about this, and actually probably the longest study on this. I think it's still going, perhaps, but I think it spanned over 100 years. And they actually took a plot in Europe where they divided the plot and they gave different inputs. One of these inputs were actually compost, and organic matter did not really change. Adding organic matter is not going to be moving your needle in terms of your organic matter. The fastest way to get organic matter into your soil are plants. That's the fastest way, and not just plants, but a diversity of these plants. And as we learned from Dan's talk earlier today, diversity of plants actually has a direct correlation, with animal health, and that should include us too, right? So yeah, if you're looking into increasing your biomass or your organic matter, diversity of plants. That's the way to do it. That's that liquid carbon pathway. So right here, we're actually on the last sample, A6, I believe. And we're at 100x total magnification. I think you probably are starting to see a pattern here, right? Seems to have a lot of silica in here, very sandy. It's somewhat sparse, which is a good sign. There goes a protozoa across the screen. Here, let's take a look at this thing here. Yeah, let's take a look. Nope. So you see how it actually starts off with, actually wait, no. It's just laying across. So that does look like a fungal hyphae. It's just that it's at different depths, right? So you can see that the one on the left, the part on the left is actually at a higher plane than the one down there. It's just a little deeper. So that's a fungal hyphae. There appears to be a fungal spore up there. Lots of bacteria here. I'm not seeing a ton of aggregation though.
Do fungal spores always appear solid, as distinguished from a protozoan spore?
No.
Which would look like an animal?
No. There's different morphologies to them, and they can be clear. With as John was mentioned earlier, with different setup, and I didn't describe this. I apologize. This is actually using bright field microscopy. There's different types of microscopy, but this is bright field, and it's a compound microscope. The reason why it's compounding is because it's compounding magnifications. You are magnifying at your eyepiece, and you're magnifying on your objective. That's what makes it a compound microscope. It's compounding these. And so with this, we're only seeing a certain spectrum of what is available to us. You can perhaps actually attach a filter to this, or even if you actually were to have a different microscope with different lighting, you would eventually end up with different views.